How is DNA evidence verified for accuracy? Imagine a crime scene where a tiny drop of blood could hold the key to solving a case. Now, think about how scientists make sure that tiny piece really points to the right person. That's what verifying DNA evidence is all about. It's a process built on strict rules and careful steps to keep everything accurate. First, DNA is collected from the crime scene. This could be blood, saliva, hair, or skin cells. Investigators use gloves and special bags to prevent contamination. Once in the lab, scientists start by extracting the DNA from the cells. Even if there's just a single hair or a tiny blood spot, modern technology can often get enough DNA for testing. Next, they measure how much DNA they have. This step, called quantitation, makes sure there's enough material to analyze. If the sample is too small, the test might not work properly. This step is important because it helps prevent false results. Then comes the process of amplification. Using a technique called polymerase chain reaction, or PCR, scientists make millions of copies of specific parts of the DNA. This is like copying a small page many times so it's easier to study. It allows even tiny samples to produce enough DNA for analysis. After amplification, the DNA fragments are separated by size using a method called electrophoresis. This process sorts the DNA pieces so they can be read. The pattern of these DNA fragments looks like a barcode unique to each person. When you see a match on screen in a crime show, it's based on this pattern. The real test of accuracy is comparing the DNA profile from the crime scene with profiles from suspects or databases. If the profiles match at multiple points, called genetic markers, it's considered a true match. Usually these markers are short tandem repeats, which are specific DNA regions that vary from person to person. The more markers that match, the stronger the evidence. But the process doesn't end there. Quality assurance is vital. Before any results are shared, another scientist reviews the entire analysis. They check everything from the extraction to the comparison to make sure no mistakes happened. This double-checking helps keep the results reliable. In complex cases, scientists might use additional techniques. For example, if the sample is mixed with DNA from more than one person, they can analyze the Y chromosome to identify male DNA specifically. Or, if the DNA is damaged, mitochondrial DNA testing can help. This type of testing looks at DNA outside the cell nucleus and can work with older or degraded samples. So, verifying DNA evidence involves collecting carefully, extracting the DNA, measuring it, copying it through PCR, separating the fragments, comparing the profiles, and double-checking all the results. Each step is designed to make sure the evidence is accurate and trustworthy. That's why DNA has become one of the most powerful tools in solving crimes and bringing justice.